How's everybody doing today? The end is near. So I have just a few things I want to say to finish up about cholesterol in the body. I think they're interesting, and understanding at least a little bit about how our body tries to regulate levels of them, of cholesterol is important. And it speaks specifically to the LDLs, because the LDLs turn out to be the only way for our body to move cholesterol into cells. The LDLs are the only way for our body to move cholesterol into cells. Now, cells can synthesize their own cholesterol, but if we want to move cholesterol in, then we've got to do it with LDLs. All right? That's why LDLs are important in the big scheme. And LDLs get into cells by, and what happens is the entire LDL gets engulfed. I think I may have shown a slide earlier in the term about that. But the entire LDL gets engulfed by the cell. And that happens because the LDL binds to a specific cell receptor that the receptor internalizes, that LDL. That brings everything in, whatever lipids are in there. The cholesterol, the fats, the fat-soluble vitamins, all those come in at the time that LDL gets brought into the cell. Okay? And it turns out that bringing LDLs into cells is a very important thing in the overall control of the level of LDLs in our body. So remember that our liver was producing VLDLs so that it could supply the cells with fat, with other fat or lipids that like cholesterol and fat-soluble vitamins. It made those and put them out into the bloodstream. Well, there's really two reasons why the liver will do that. One is because the body needs it, and the other is because the liver is just so full of this stuff, it's got to do something with it. This is where diet and obesity can be a factor in cholesterol. Too much capacity for the liver to hold stuff, the liver's going to start dumping it. Okay? Now, you hear these stories about people who go out and they eat a dozen eggs every day for breakfast, and they eat a horrible diet, and their cholesterol levels are perfect. And you think, well, how can that be? And that can be because they have some things going for them that I'm going to describe to you. All right? So some of those things that are going for them are really a good monitoring system in their body. So I'm going to describe for you what that monitoring system is all about. All right? So the monitoring system occurs in your liver. And your liver has a special LDL receptor that grabs LDLs out of the bloodstream. Why does it do that? Well, it does that as part of this balance that's out there. And it does it partly to determine, does the body need more VLDLs? Okay? So, the liver has sent out a bunch of VLDLs, and it knows how much it's sent out, and now it knows how many LDLs are coming back into it. And it looks at that and says, whoa, the body is really gulping up these VLDLs I'm putting out. I should put out some more. And it measures that by the amount of LDLs that make it back. Okay? The more LDLs make it back, the liver says, oh, wow, this person is not taking up all that I have put out, so I'm going to stop putting VLDLs out. Okay? So the level of LDLs in the blood in an ideal person is, in fact, being read by the liver and is a determinant of how many VLDLs will get put out there eventually. Okay? So when that system is working and working well, then you really can do a lot with your cholesterol and eat a lot of cholesterol and all these various things, and your body will manage it just fine. Okay? Now, people who are overweight or who have liver problems may have other issues with that recognition, because if there's anything that screws up the recognition by the liver thinks, oh, wow, there's no LDLs out there. I should be putting out even more VLDLs. All right? I had a rather bizarre story. It was a true story that happened when I was teaching this class about 10, 12 years ago. it wasn't this class, it was another class. But in any event, the student came up to me after class, and there's a disease called familial hypercholesterolemia. And in this disease, people who have the disease have a genetic problem such that their liver receptors, the special receptors for the LDLs, are defective. So when that happens, then the LDLs make it back to the liver, but the liver never sees them because there's no receptor for them, and the liver says, oh, wow, I got to put out some more VLDLs because this guy's taking up everything I'm putting out. So it starts dumping as much as it can into the bloodstream. Well, people who have this disease usually die by the time they're about 10 years old because they die of a heart attack because their blood just gets all plugged very, very quickly with this stuff. So anyway, this lady comes up to me and she says, my son's got that. And I said, there's no way your son has that. First of all, it's a very, very rare genetic disorder. It doesn't happen very often. And number two, how old is your son? And she says, he's about 12. And I said, that's a pretty fatal disease fairly early. And she says, no, I'm sure that's what he's got. And so I said, okay. So she goes home and she brings back and she has a picture. And she shows me the leg of this kid. And the leg has these great big bubble-like things on his leg. And I said, what the heck is that? And she says, well, they said it was cholesterol. You're kidding. Well, it turned out the kid did, in fact, have hypercholesterolemia. And they discovered it, fortunately, when he was about 4 or 5 years old through a routine blood check. All right? A person who has what we call high cholesterol, we start worrying about cholesterol levels when it gets up to about 200 in the measure in the bloodstream. Okay? This kid had a measure of 700. 700, okay? Well, how was it that he was able to live and go through all this? Well, at about that time, the statins were just coming out, and other cholesterol drugs that controlled levels of cholesterol were starting to come out, and they had put this kid onto it very early and very quick, and they had gotten his cholesterol levels down to about 200. So it looked like he was actually going to live a fairly normal life in spite of lacking this receptor. A pretty remarkable story, but absolutely true. But familial hypercholesterolemia, I'll put it in I won't spell it here, I'll put it into the highlights, is a genetic disorder that happens because of that lack of LDLs on the liver. Interesting disorder. Okay, that's the last of what I want to say about cholesterol. I think I'm going to turn our attention, unless there's questions, I will turn our attention to photosynthesis, which I know some people are anticipating readily. So let's talk about photosynthesis. And this is a great day to talk about photosynthesis. The sun is out there doing photosynthesis left and right. It's also doing sunburns left and right based on what I've been seeing of people. All right. Well, photosynthesis is a process that occurs inside of the chloroplast. And the chloroplast has an anatomy like you see here. The chloroplast is somewhat comparable to the mitochondrion, as we will see, but the chloroplast has more elaborate structure on the inside of it than the mitochondrion does. We are going to be very concerned with the thylakoid spaces because that's where a lot of the action of photosynthesis is going to occur. The thylakoid spaces, back up and say this. We saw in the mitochondrion that the way that ATP was made in the body was largely by means of pumping protons out of the mitochondrial matrix into the intermembrane space, and then those protons came back in, and when they came back in, that's how the gradient was formed. Okay, that's how the ATP was formed. That gradient was what enabled that to happen. Okay, when we look at chloroplasts, okay, instead of seeing things being pumped out, we see things being pumped in. So the gradient's in reverse. We have high concentration of protons inside of these spaces in here, the thylakoid spaces, and it's the movement of protons out that causes ATP to be made. It really doesn't matter as long as we orient everything properly so that the ATP is in fact being made as a result of the movement of those protons. Okay. I'll come back to that. Okay, so as we talk, the specific structures inside of the chloroplasts that capture the energy of the light of the sun are, as I'm sure everybody knows, chlorophyll. And chlorophyll, as I've mentioned previously, has a structure, the active center of chlorophyll has a structure that's almost identical to that of hemoglobin. It has a porphyrin ring. The main difference is right there in the middle that you can see. The main difference is that instead of having an iron at the center of it, it has a magnesium at the center of it. Okay? Chlorophyll has long nonpolar tail for anchoring it into the membrane of the thylakoid, as you can see here. And long nonpolar tails are very good for that purpose, as we have talked about before. Okay. Now, when we talk about photosynthesis, we talk about photosynthesis as occurring in really two phases, a light phase and a dark phase. The light phase requires light. The dark phase does not require dark. Okay? Common misconception. So the dark phase does not require dark. The dark phase simply does not require light. There's no light energy needed to power the dark phase of photosynthesis. Okay. Well, in the light reactions, we're going to spend some time talking about those. And what I do with photosynthesis is I try to keep it as simple as I possibly can, simply because the names and the movements of all of the electrons in photosynthesis are rather complex. And for our purposes, I don't think it's necessary to memorize them. Okay? We will understand the process as a whole, and when I'll point out to you the highlights of the movement of those electrons, but the individual, this goes to this, goes to this, goes to this, we're not going to concern ourselves with. Okay. So, light energy is harvested by chlorophyll in what are called reaction centers. And reaction centers are sort of as you depict here. You can see energy being grabbed by one, and it's sort of passed along the reaction centers ultimately, or along the various harvesting pigments until they get to what a reaction center actually is. It's at the reaction center that the reactions I'm going to talk to you about will actually occur. So we can think of this almost as an electronic network that is communicating that information to the place where the reactions will occur. Okay? Now, there are different types of photosynthesis that occur in different kinds of organisms. We're going to talk about photosynthesis in very general terms, and we're going to talk about it in terms that occur in plants. Bacteria that do photosynthesis, some bacteria do photosynthesis, have some of the reactions that we see here, but not all the reactions that we see in photosynthesis. Photosynthesis occurs in two different photosystems. These are the reactions now that are occurring in that reaction center. Two different photosystems. And the first one that the reactions occur in is known as photosystem 2. Logic, right? Okay. So, what happens during photosynthesis? Well, we have what's called a Z-scheme. And I'm just going to sort of step you through the Z-scheme right now, and then I'll come back and look at it a little bit more closely. The Z-scheme is drawn to show energy changes, and it's drawn to show energy changes and transfer of electrons between two different photosystems. So we see two photosystems that are working together to do the overall process of photosynthesis. Photosystem 2 is where the process starts. Photosystem 1 is where the process continues. Both steps are needed for the full cycle of photosynthesis. Okay? Both steps are needed for the full cycle of photosynthesis. In photosynthesis, we are doing, in many ways, the backwards of what we were doing in oxidative phosphorylation and electron transport. If we think about electron transport, what was the final source, what was the final destination of the electrons? Oxygen to make water, right? Well, in photosynthesis, the very first reaction occurs in which oxygen is taken from water. So water is done what we call split, and that splitting of water generates molecular oxygen. It's that splitting of water by the process of photosynthesis that actually is responsible for the vast majority of oxygen that's on Earth today. We talk about the fact that in early in Earth's history, the planet had a very reducing atmosphere. It had very, very little oxygen. But by the time photosynthesis evolved, then we began to see production of oxygen into the atmosphere. It gave us enough oxygen, obviously, here today to breathe and to live. So we're dependent upon plants for oxygen generation. We're also dependent upon plants for all the oxygen that's been here over the eons. Okay, so that's a schematic. We see electrons moving, we see electrons moving, we see electrons moving, and finally we see an electron acceptor, because we know that if electrons move, they have to go somewhere, because electrons, if they just attach to any given atom, can cause that atom to become very reactive, and that's not good. All right? Well, as I said, this is the sort of backwards of the process of electron transport, because the final electron acceptor in photosynthesis is NADP. Along with a couple of protons, it makes NADPH. In electron transport, NADH was the source of the electrons. In this case, NADP is the acceptor of the electrons. So we're kind of running this process in reverse. Well, how is that possible? Because in electron transport, that process was made possible by virtue of the fact that those electrons flowed freely. The electrons started at a high energy state, and they traveled lower, 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 lower until they finally got to oxygen. If we're going to reverse that process, how do we make the electrons climb that energy ladder? Well, the energy ladder is climbed with these two steps. Light is providing the energy necessary to get electrons backwards, as it were, in the overall process. That's a very, very important consideration. All right? So, what I've just described to you are the main features of the process of photosynthesis. Oxygen evolution from water, okay? Movement of electrons, accepting of the electrons by NADP, and the whole process is driven by light. Okay. Now, this, from a perspective of the membrane, is what is actually going on. So here's the lumen of the thylakoid. There's the stroma. That's the outside part, the area outside of the thylakoid. And here's what's happening. All right? We have water being split into protons plus molecular oxygen. Okay? Well, how does that happen? That's the very first thing that we have to consider. This is a process that a lot of people would really love to be able to harness in various cells, or not living cells, but cells that we construct. Because this splitting of water can be used to generate, if we do it right, hydrogen. And hydrogen is a very good energy source for us to burn. All right? Well, the way that the plant accomplishes this, and we're at a reaction center right now, the way that the plant accomplishes this is that there is a complex that exists where this whole process starts. And this complex has a manganese atom as part of it. A manganese. Not a magnesium, but a manganese. This is a complex that has manganese. The capturing of the light is coming by the magnesium in the chlorophyll. The actual splitting of the water is happening in this manganese complex. So how does this happen? Well, there is a photon of light that comes in, and the electrons that were associated with that chlorophyll get excited. And they are really, truly excited, meaning they go to a higher energy state than they were at before. So electrons, by the energy of light, are excited to a high energy state, and that causes them to leave the chlorophyll. Okay? The electrons are leaving. Well, when you leave electrons, that leaves behind a hole. And the hole occurs at the manganese complex, which gives up its electrons to make up the whole, but now the manganese complex has no electrons of its own. It looks for a source of electrons, and the nearest source of electrons comes from molecular water. Okay? It's coming from molecular water. It pulls those electrons away, and it's the pulling of those electrons away that results in the generation of molecular oxygen and 2 protons. So, to recap what I've just said, light energy comes in, excites electrons in the chlorophyll, removes those electrons, leaving a hole. The hole is filled by electrons from manganese, which is left with a hole of its own, which grabs electrons from water and generates molecular oxygen and protons. This is all the very beginnings of what we call the light cycle. And not surprising, because light is necessary to drive this process. Well, what do we have? We see a big complex, we see a big complex, and we see a big complex. What did we see inside of the mitochondrion? We had complex 1, we had complex 2, we had complex 3. These guys have different names, but they're big complexes that are located inside the membrane of the thylakoid. Okay? Well, those complexes don't move very well, just like the big complexes inside the mitochondrion don't move very well. Complex 1, you recall, gave its electrons to coenzyme Q. Coenzyme Q is a little tiny molecule that moves back and forth very easily. Okay. Complex 3 gave its electrons to cytochrome C, which is also a little tiny protein that moves back and forth very readily. So those big complexes in the mitochondrion are donating electrons to littler things that can move readily, and that's exactly what's happening here in the membrane of the thylakoid. This big complex donates its electrons to this thing called PQ, and no, by the way, you're not going to need to know that. PQ stands for plastoquinone. And no, you don't need to know that either. This guy's shuttling electrons. It carries electrons from this complex over to this complex. Electrons travel through this complex, and look what's happening during the process. As they travel through this complex, we see protons being pumped inwards. That's the backwards direction from what we saw before. Protons are being pumped inwards. Okay? This complex now, which passes some electrons off to another small molecule, plastocyanin, PC, you don't need to know that, transfers its electrons over to this big complex, and this big complex now is the beginnings for photosystem 1. Photosystem 1 accepts energy from the sun, takes those electrons that have been donated to it, and excites them to a high level, where it gets dumped to something called ferredoxin, FD, right there. Okay? Ferredoxin donates its electrons finally to this complex, a flavoprotein that donates electrons to NADP and makes NADPH. I don't care that you know the names of the intermediates in here. No. Okay? The end product of this was now we've made NADPH, and NADPH, you may recall from our talk about the synthesis of fatty acids, NADPH is very, very useful for anabolic processes, for making things. So now the cell has a great source of NADPH. In animals, who remembers where we get most of our NADPH from? Not from pyruvate. Penose phosphate pathway. That's right. Penose phosphate pathway is our primary source of NADPH. In plants, here's their primary source of NADPH. Okay? Now, as the process of all this stuff going on, what's happening? Well, protons are getting pumped in. That means we've got a proton gradient, meaning that there's more protons in here than there are out here. And those protons want out, and exactly like what happened in the mitochondrion, as the protons move through this last ATP synthase, which, by the way, looks almost identical to the one in the mitochondrion, as they move through this ATP synthase, ADP is converted into ATP, and we have, as a consequence, ATP being made. Now, this process overall, in addition to being called photosynthesis, is called photophosphorylation. Photophosphorylation. Earlier in the term, I told you that there were 3 types of phosphorylation. They were known as substrate-level phosphorylation. That occurred in glycolysis, for example, where 1,3-BPG donated a phosphate to ADP to become ATP. The primary source of ATP that we've talked about before this was actually in oxidative phosphorylation. That's the second type. And the third type is photophosphorylation. Photophosphorylation, of course, is what's occurring only in photosynthetic organisms. Okay. Now, I give a lecture that one of the more popular silly ideas that I give to people, but some say it's not so silly, is the idea of how to make a photosynthetic fish. Would you like to hear how to make a photosynthetic fish? Has anybody ever heard it? You've heard my lecture on that? Okay. A photosynthetic fish is actually not as difficult to make as you might think. All right? All right. So, what you've learned so far about proton gradients is that proton gradients can be used to make ATP, right? If we have an energy source, or we have a thing or complex that can capture the energy of light, we can make ATP if we set it up right. Okay? And that's what I intend to do in making a photosynthetic fish. Okay? How do we do it? Well, first of all, we have to have a fish that we can get light into. You ever seen those little glass fish they sell in the aquariums that you can see through? This is what's going to be my photosynthetic fish. I'm going to genetically engineer this fish so that it can make its own ATP. And I've talked to people about this idea, and they say it's not completely crazy. I'm crazy, but the idea is not. What's that?
There's a photosynthetic sea slug. It steals plastics.
There are sea slugs that, in fact, do steal chloroplasts from certain plants and use them. But I'm actually talking about a fish that can do this, that has it as its own. Okay? So how does that happen? All right? Well, there's a very interesting protein that's found in bacteria called bacterial rhodopsin. And bacterial rhodopsin has a very interesting property. In bacteria, it does the following thing. Okay? It's got a sort of a chamber, and it's a little tunnel. And in the middle of that tunnel, there's a molecule of vitamin A. And vitamin A is a light-sensitive vitamin. That's what gives us vision, for example. And we get vision as a result of signaling that happens with vitamin A, because in the presence of light, vitamin A has some double bonds. It's got a side chain. It's got some double bonds. Those double bonds can flip from the cis configuration to the trans, meaning it can go from straight to bent, straight to bent, straight bent, straight bent. You put it in light, and that's what happens to vitamin A. Well, bacterial rhodopsin has this protein in the middle of this chamber. It's got a molecule of vitamin A. And when light hits bacterial rhodopsin, that vitamin A goes bent, 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 like that, right? Bent, unbent, bent, unbent. And each time it goes through bent, unbent in the bacterium, what it does is it grabs a proton and it kicks it down out the chamber. That's what it does. So in the bacteria, they're using this bacterial rhodopsin not as a way of seeing or having vision, but instead as a mechanism of pumping protons. Start to see where this is going? For a photosynthetic fish, all we really need to do is take and put the gene for bacterial rhodopsin in the fish so that the bacterial rhodopsin is made in the mitochondrion of the fish. And what's going to happen when you do that is when you shine light on Mr. Fish, a proton gradient will be created. And when that proton gradient is created, what happens? It comes back through ATP synthase, and it makes ATP. You have a photosynthetic fish. Okay? I want a cut of whoever makes that fish. All right? That's how you make a photosynthetic fish. Okay. So, it doesn't take a plant. It doesn't take a snail stealing the chloroplast, which is what it's doing, of plants, or using them sort of symbiotically, which is also part of what it's doing. Instead, you can genetically engineer this fish and make it make ATP. Now, it won't be like a plant in the sense that the plants have a dark cycle to their photosynthesis. The dark cycle is where they take carbon dioxide out of the atmosphere and use it to make things. The fish doesn't have that. All right? So the fish would still need to have a carbon source, but it wouldn't need to eat nearly as much as a regular fish because whatever carbon you gave it, it was going to use with its own energy to make ATP and burn things. So it would only use carbon as a way of building molecules. It would not use carbon as an energy source. Anyway, okay. For what it's worth, that's my photosynthetic fish. All right. Well, this completes what happens in the light cycle of photosynthesis. Okay? This is the light cycle of photosynthesis. All right. Here are some of the molecules that play roles in that process. You'll see they look not unlike coenzyme Q. They have long tails that allow them to be embedded in the membranes. Here are the electron-carrying portions out here in the ring, okay, or this ring. All right. Here's some other quinones that are involved, and you'll see they have multiple rings in some cases, single rings in others. But these things are important for carrying electrons during that movement of electrons from complex to complex to complex as they're traveling through in photosynthesis. Okay. Well, not all photosynthesis is the same. And in fact, there is photosynthesis that occurs in plants that deviates from the process I've just shown you. Not all photosynthesis goes through that last process that I just described to you. Okay? Well, what's happening here is something called cyclic photophosphorylation. And it's a way of bypassing what was happening in the earlier cycle. This is happening mostly through complex 1 and bypassing some of the things in complex 1. Okay? All right. What's happening is we see complex 1 is getting exciting electrons, but the difference here is that there's no place for the electrons to go in the standard scheme. In the standard scheme, they would go on to NADP to make NADPH. But what's happening in every plant out there in the quad right now is the fact the sun's been shining on them all day long, and cells have limiting amounts of NADP. Just like you have limiting amounts of NAD and NADH, plants have limiting amounts of NADP. They've gotten so much sunlight that all of their NADP is in the form of NADPH. Well, instead of allowing all of that energy that's coming down and doing no good, it would make no sense to just stop the process because we've run out of, or because the plant has run out of NADP. Instead, there's a short circuit that occurs. The short circuit occurs when electrons, instead of being passed off to NADP, get passed backwards, okay, through this complex here. This complex is therefore still allowed to pump protons into the thylakoid. So we're short-circuiting photosystem 1 and passing electrons backwards through this proton pump to pump protons into the thylakoid space. Light energy can continue to be harvested, protons can continue to be pumped, and ATP can continue to be made. That's the advantage of cyclic photophosphorylation. Without this short circuit occurring, the lack of NADP would stop the plant from being able to make ATP. Okay. Now, the plant is still going to need ADP, just like we're going to need ADP. All right? But as long as it's got ADP, it can keep this process going through this short circuiting that I'm describing to you here. Okay. There's all the players. I don't think you want me to go through all that again. Okay? And when we look in bacteria, photosynthetic bacteria, we see that they use something like photosystem 1, the cyclic photophosphorylation. They don't have two complete photosystems. They're using something very much like the photophosphorylation that I just described to you. I shouldn't say we do. We don't do that. But that plants do. And they also, as a byproduct of that, make NADP. That's a little bit more involved than the cyclic photophosphorylation. But nonetheless, they only have one photosystem for doing all this, and they're still making ATP in the process. Okay. Those are the light reactions that I want you to know. Now we turn our attention to the dark reactions, and as I said, the dark reactions don't require dark, they just don't require light. Right? It's in the dark reactions of photosynthesis that the It's in the dark reactions of photosynthesis that all of the energy that is captured in the light reactions is used. And it's used to do something that's also critical for stability on Earth, and that is for the assimilation of carbon dioxide. Okay? We all hear about how global warming is resulting from the greenhouse effect, and the greenhouse effect is arising from the fact that carbon dioxide concentrations in the atmosphere are increasing. And one of the things that decreases carbon dioxide in the atmosphere is actually the dark reactions of photosynthesis, assimilation of carbon from the atmosphere. Unfortunately, we're burning it. We're burning carbon to make carbon dioxide faster than our plants and other organisms can assimilate that. All right? But without those, we'd be in even worse shape. Well, the dark reactions of photosynthesis come in through a complex process called the Calvin cycle. Yeah. The Calvin cycle is shown on the screen, and I'm going to point out some highlights in the Calvin cycle to you. All right? I'm not going to go through the entire Calvin cycle. We could, but we're also not a majors biochemistry course. All right? Nevertheless, the Calvin cycle has some important things that we need to understand. All right? One of the important things we need to understand in the Calvin cycle is this very first reaction. The very first reaction of the Calvin cycle involves the capture of carbon dioxide from the atmosphere by the plant. Okay? In this reaction, what happens is a molecule known as ribulose 1,5-bisphosphate, and you can call that RUBP if you wish. That's a 5-carbon molecule. That 5-carbon molecule is combined with carbon dioxide from the atmosphere to make 2 molecules of 3-phosphoglycerate. So we've got 1,5 plus 1 carbon, 1 5-carbon molecule plus a 1 carbon dioxide, giving us a total of 6 carbons. That produces 2 molecules of 3-phosphoglycerate. Okay? Now, you'll see some other numbers up here. You see 6, and you see 6, and so on and so forth. Those are simply to balance the overall equation. And if you want to balance the overall equation, you can. You'll probably get very confused as you go through. We're not going to worry about balancing the equation. So, the process starts with the assimilation of carbon dioxide from the atmosphere in this reaction I've just described to you. And it's catalyzed by a very, very interesting and important enzyme. This enzyme, in fact, has been described as the most abundant enzyme on the face of the earth. It's called ribulose bisphosphate carboxylase, which is a mouthful of a name. You can call it Rubisco, R-U-B-I-S-C-O. Rubisco catalyzes this reaction, and interestingly, this enzyme turns out not to be very efficient at all. It turns out not to be very efficient at all, as we'll see in a little bit. There are some byproducts that it can produce, and it's not completely understood why this enzyme has evolved to this point of being so very inefficient that it is. All right. Well, let's just step through. The next steps I'm going to take you through for the next couple of minutes, you're not responsible for. But I just show these to you to let you think about how these reactions, many of which you've already seen before, actually overlap with each other. All right? 2 molecules of 3-phosphoglycerate are converted into 1,3-bisphosphoglycerate. Does that sound familiar? It sounds like gluconeogenesis, doesn't it? 1,3-bisphosphoglycerate gets converted into glyceraldehyde 3-phosphate. Yep, that sounds like gluconeogenesis, which also gets converted into dihydroxyacetone phosphate. Yep, that sounds like gluconeogenesis. And in fact, a lot of the reactions that we see here sound like gluconeogenesis because the end product of this overall process is glucose. It's right there. The reason you saw 6s up here was it takes 6 carbon dioxides to make the 6 carbons that go into 1 molecule of glucose. We talked we talked recently about the pentose phosphate pathway. And I said it was an important source of NADPH, and I showed you how the pathway, you could go from taking a 3-carbon sugar and a 7-carbon sugar and make 2 5-carbon sugars. All right? If we look at the reactions that are occurring in the Calvin cycle, there's a 7-carbon sugar, there's a 4-carbon sugar, these are 3-carbon sugars, and they're making a 6-carbon sugar. The Calvin cycle has many of the same reactions as gluconeogenesis and the pentose phosphate pathway. Those processes are very, very related to each other. Well, the upshot of this is that we have used ATP and we have used NADPH in the process of making glucose. And where did those come from? Well, those came from the reactions that were producing them in the light cycle of photosynthesis. Okay? So consequently, this overall process is happening driven by light, even though it doesn't itself require light. Okay? The energy sources for the dark reactions come from the light reactions. Okay. That's pretty cool. Let's, here's the fixation reaction. So here's the ribulose 1,5-bisphosphate. Here's a big intermediate that you don't need to know. All right? And then this big intermediate falls apart, and when it falls apart, you get 2 molecules of 3-phosphoglycerate. Okay. This is what Rubisco looks like. Live and in color. Question? Okay. And this is a description of it. It's probably the most abundant protein on Earth. Okay? Every plant is loaded with Rubisco. All right. Now, we hear different things about, what am I doing on time? Oh, I've got 5 minutes. So we hear different things about different types of plants that are out there called C3 plants and C4 plants. Botanists definitely know what I'm talking about here. And C4 plants are different fundamentally from C3 plants in the way that they do the capture reaction for carbon dioxide. Okay? I'll repeat that. The C4 plants differ from C3 plants primarily in the way they do the capture reaction for carbon dioxide. The scheme I just showed you in the dark cycle, I didn't say where that carbon dioxide came from. I said it came from the atmosphere, and it's true. The carbon dioxide for the Calvin cycle comes from the atmosphere for both C3 and C4 plants. Okay? They're both sucking carbon dioxide out of the atmosphere. All right? But in a C3 plant, they capture that directly, that carbon dioxide directly and do the reactions of the Calvin cycle directly on the surface or right at the very outermost part of the leaf. That turns out not to be the best place to do it. And the reason it's not the best place to do it is if you want to get carbon dioxide into the leaf, you have to have relatively porous leaves. And if you have relatively porous leaves, you're likely to lose a lot of water. Okay? Losing water for a plant is okay if you have plenty of water, but if you don't have plenty of water, then you've got some problems associated with that. So it's probably because of these conditions that C4 plants evolved a different mechanism of getting their carbon dioxide to the Calvin cycle. How does that happen? Well, in the C3 plants, as I said, it's going on out here in the outer part of the leaf. They grab it onto the ribulose 1,5-bisphosphate, and they do the Calvin cycle right there. C4 plants, on the other hand, capture carbon dioxide for the very first time not on ribulose 1,5-bisphosphate, but instead on phosphoenol pyruvate. They convert PEP into oxaloacetate in the first reaction. Now, why are they doing this? Well, they're doing this because they can capture that carbon dioxide here with less permeability for water getting out. They can take small quantities of carbon dioxide and assimilate this more readily. Then they take and they move those carbons further into the plant where water is readily available, where there's less loss and so forth of the water, and they have the Calvin cycle going on internally. So these plants take oxaloacetate, they convert it to malate, they then transport it further inside of the plant, and inside the plant the carbon dioxide is donated to ribulose 1,5-bisphosphate, just as we saw before. So C4 plants produce 2 molecules of 3-phosphoglycerate, just like C3 plants do. And they use ribulose 1,5-bisphosphate, just like C3 plants do. The difference is where they do it. In the case of the C3 plants, they're grabbing the carbon dioxide directly with ribulose 1,5-bisphosphate. In the C4 plants, they're grabbing it with PEP and then donating it to ribulose 1,5-bisphosphate inside of the plant. This is a messenger. This messenger system is delivering carbon dioxide internally. The result of this is that C4 plants turn out to be much more efficient in the process of photosynthesis than C3 plants do. They've got a concentrating mechanism for getting that carbon dioxide in, and it works very well for them. Okay. Now, there are their individual reactions. You don't need to worry about the individual reactions, but that's what's happening in the C4 plants. The last thing I want to say here, actually, I'll save the last thing for tomorrow. I do have a song for you, and it's all about photosynthesis. It's to the tune of Scarborough Fair. Photosynthesis is divine, fixing carbon using sunshine. It's thanks to plants that we've got a prayer. They pull CO2 from the air, reaping energy from the sun. It's efficient, second to none. You grab the photons almost at will. Protoporphyrin chlorophyll. Light reactions of system 2. Split up water making O2. Electrons pass through schemes labeled Z, pumping protons gradiently. ATPs made due to a shift of the proton spinning quite swift. An enzyme turbine cellular maze, you know as ATP synthase. Carbons fixed onto a substrate. Ribulose 1,5-bisphosphate. Rubisco acts inefficiently, splitting it into 3 PGs. If the enzyme grabs NO2, it makes glycolate, it is true. The Calvin cycle works in a wheel, giving plants a sugary meal. So photosynthesis is divine, 'cause it happens all of the time, from dawn to dusk and times in between. Solar panels truly are green. All right.